Hello everyone. Very good evening and welcome to this week 6 NPTEL live TA session for the course Cell Culture Technologies. My name is Ankita De and I'm one of the TAs for this course, and I host this session every week on Tuesdays 7 to 8 p.m. So let's get started. So, as you already know, this is a 1 hour live interactive video session where we discuss some of the previous year's assignment questions of the course Cell Culture Technologies, which help us in solving this year's assignment questions. And towards the end of our session, we have a Q&A segment where we discuss some of the previous year's assignment, where if you have any queries, you can post them in your chat box and I try to solve them. Also, at the end of each session, we provide you with the course materials and the class recordings for your access later. Let's get started. So before we move on to discussing the assignment questions, we will quickly recapitulate some of the key points that were discussed in this week's course lectures. So in this week's lectures, we had discussed about the different types of cells that are found in the central nervous system, the CNS, and the peripheral nervous system, the PNS. Then we will also try to understand through our slides the mechanism of the nerve impulse transmission across a synapse. Further, we have seen or discussed in this week's lectures about the primary co-culture of the neuronal and non-neuronal cells in vitro and how it can help us in understanding the mechanism or the basis of several neurodegenerative diseases. Then we have seen a method or the procedure how we can isolate the DRG or the dorsal root ganglion from the rat embryo. Next, we have discussed about the several techniques, so the cell separation techniques, which are the mechanical, enzymatic, density-based, and the immunosorting techniques by which we can separate the several neuronal and the non-neuronal cells from each other. So we will discuss what each of these are in our upcoming slides. So let's move on to our next slide. So in this week's lecture, we have discussed about the different cells that are found in a nervous system. The nervous system is composed of different types of cells. So they could be the neuronal cells or the non-neuronal cells. So the non-neuronal cells are so all these cells are referred to as the neural cells. So neural cells could include both the neuronal cells as well as the non-neuronal cells. The non-neuronal cells are the cells which are mainly associated with providing growth, support, and myelination to the neuronal cells. And the neuronal cells are mainly associated with the transmission of the electrical signal or the transmission of messages across the nervous system. The neuronal cells are referred to as the neurons, and the non-neuronal cells could be of several types. So we will discuss what each of them are in our next slide. So the non-neuronal cells are the glial cells, and then we have the neurons. The neurons are responsible for sensing a change in the environment and communicating with other neurons via the electrochemical signals, while the glial cells are present to support, nourish, and insulate the neurons and remove the waste products of metabolism. So this is how a typical neuron looks like. It's made of several components. So you have already seen how the structure of a neuron in some of your early classes. So this is the cell body or the soma. So this is composed of the nucleus and several other intracellular organelles such as the mitochondria, the Golgi apparatus, also the Nissl granule, which are the sites of the protein synthesis because it contains the free ribosomes, and the rough endoplasmic reticulum. Next, we have the dendrites, which are the projections. These projections which project outward from the cell body or the soma. So the dendrons, or the dendrites are responsible for receiving a signal from other neurons or cell body. Next, we have the axon. This is this long projection that originates from a region of the cell body which is known as the axon hillock. So the axon is responsible for transmitting the signal away from the cell body towards the synapse. Next, we have the Schwann cells, which are not shown here. So the Schwann cells are mainly responsible for myelinating the axon, and Schwann cells are responsible for myelination in the peripheral nervous system. Next, we have the axon terminal over here. The axon terminal is responsible for transmitting the signal to other neurons with the help of several neurotransmitters. So next we have the glial cells, which are the non-neuronal cells. So the glial cells could be of different types. So we have the astrocyte, the oligodendrocyte, microglia, and the ependymal cells, and each of them have a different function. So the oligodendrocytes, as I've already discussed, the oligodendrocytes are responsible for myelination in the central nervous system, while the Schwann cells are responsible for myelination in the peripheral nervous system. So we will discuss about the function of the astrocyte. So these are the star-shaped glial cells which are found within the brain and the spinal cord, and they have numerous functions that include providing metabolic support, regulation of the extracellular ionic environment, neurotransmitter uptake, modulation of synaptic transmission, and the promotion of myelination by the oligodendrocyte. Next, we have the oligodendrocytes, which are responsible for insulating the axons in the central nervous system. So they carry out this function by producing a myelin sheath which enwraps around a part of the axon. A single oligodendrocyte can myelinate up to 50 axonal segments. Next, we have the microglia, which are the resident immune system of the brain. So the microglia make up about 10 to 15% of the cells within the brain and are of a mesodermal origin, unlike the glial cells which are of ectodermal origin. So they are activated in response to tissue damage and have the capability to recognize foreign antigen and initiate phagocytosis to remove the foreign material. Last, we have the ependymal cells. The ependyma refers to the lining of the ventricular system of the brain and the spinal cord. This lining is made up of the ependymal cells, the basal membrane of which are attached to the astrocyte. And the main function of the ependymal cell is to produce cerebrospinal fluid as a part of the choroid plexus. So these are the four main types of cells which compose our glial cells. Next, we will discuss about the mechanism of the nerve impulse transmission across a synapse. So when a nerve impulse transmission takes place, we have a synaptic cleft region. So when a neuron receives a signal from some other neuron, then it will transmit the signal to the end of the axon terminal. The axon terminal in turn has these synaptic uh, bulb region, at which we have this presynaptic knob-like region, which is a swelling at the end of an axon terminal. So this swelling or the swollen region contains several vesicles, which in turn contain neurotransmitter molecule. So these neurotransmitter molecules are responsible for carrying the signal across the synaptic cleft region to the receiver muscle or the neuron. So there is a synaptic cleft in this region, which is for where if the signal needs to pass from this neuron to this neuron, it has to be transmitted by the help of these neurotransmitter molecules, because there is a gap here. And the post-synaptic, this could be a post-synaptic neuron, or it could also be a muscle. So the transmission could either take place from a neuron to a muscle, or it could take place from a presynaptic neuron or to a postsynaptic neuron. Now, how does this action takes place? So the incoming signal is transmitted when one neuron releases a neurotransmitter into the synaptic cleft, which is detected by the second neuron through the activation of receptors placed opposite to the release site. So in step 1 is the nerve impulse reaches the end of the axon terminal. Here we have several neurotransmitter containing vesicles. So when the nerve impulse reaches the site, the voltage gated calcium ion channels present here will open. This will result in the movement of calcium ions from the extracellular space to the inside of the axon terminal, or to the inside of this bulb. This in turn will allow these neurotransmitter containing vesicles to go and bind, or rather fuse, to this membrane. And on fusion of these neurotransmitter containing vesicles to these membranes, they will release these neurotransmitter molecules into this region, this gap region, which is also known as the synaptic cleft. So after the release of these neurotransmitter molecules into this cleft like region, these molecules will go and bind to the specific receptors on the surface of is ligand-gated ion channel on the surface of the postsynaptic neuron or the muscle. So on binding of these neurotransmitter molecule on these postsynaptic neurons, they will open these ligand-gated ion channel, which will result in the inflow of sodium Na plus ions that will result in another depolarization or a nerve impulse or a new nerve impulse, which will then be carried along the length of the next neuron or the second neuron. So this could either be a muscle or a neuron. So this is basically how nerve impulse transmission takes place across a synapse. Next, we have talked about in this week's lecture about how primary in vitro culture of murine or the rat embryonic dorsal root ganglia isolated from the spinal cord and their co-culture with Schwann cells could act as a model or serve as a test bed to understand the basis of several demyelination associated neurodegenerative diseases. So we have discussed that after we isolate the cells via the mechanical disaggregation and the enzymatic action, it is important that we segregate and characterize the different cell types. So in connection to this, we have discussed about the different sorting or the segregation processes. We have discussed about the two important techniques, the differential density gradient centrifugation and the fluorescence activated cell sorting methods. So we will discuss about them in our upcoming section. So in this week's lecture, we had discussed about how we can isolate the dorsal root ganglia from the murine spinal cord. So this is how the dorsal root ganglia look like. So as you can see in the journey figure, the dissected dorsal root ganglia can be dissociated and grown in culture or section for immunohistochemical analysis. The figure A shows that the DRG or the are the clusters of the sensory neuron cell bodies located in the dorsal root of the spinal column. The schematic portrays a transverse section of the spinal column. So the mice possess 8 cervical, 13 thoracic, 5 or 6 lumbar and 4 sacral DRG pairs totaling 60 or 62 individual ganglia depending on the genetic background. So this picture depicts the dorsal aspect of the spinal cord and the spinal nerve roots without bilateral DRG for simplicity. See, and these are C and D represent some of the primary DRG sensory neuron cultures 24 hours post plating. In the lecture videos, we have discussed how we can isolate the dorsal root ganglia from the murine spinal cord. So I could not find an appropriate video which describes this process, but I will try to provide you if I find them in the slides that I in the PPT slides later. So next, as I was discussing that how we can separate these cells from each other that we are dissociated from the dorsal root ganglia. So the dorsal root ganglia might contain different types of cells. So they could be of the neuronal or either of the glial cell origin. Now it is important that we separate these cells into different fractions so that the glial cells that we have, which are which are the dividing cells, do not overpower or take over the entire surface of the culture plate, which in which we are culturing our neurons or the neuronal cells. Because as we know, the neuronal cells are the non-dividing cells, and they do not proliferate. They might show some neurite outgrowth, etc. But however, they do not proliferate in culture and we can expand them only up to one or two cell divisions only. So it is important that we separate the glial cells and the neurons from each other. So this we could do following two procedure, and one of them is shown here, which is the density gradient centrifugation method. The first step in fractionating the cells from the neural tissue is to release them by the enzymatic disaggregation process. So we have discussed about the mechanical and the enzymatic disaggregation process in the previous lecture video. How we, with the help of a scalpel and a needle, can separate the dorsal root ganglia from the spinal cord, and then with the help of enzymatic action, that is with the help of an enzyme papain, which is a less harsh enzyme compared to to its other counterparts such as trypsin, we can separate the cells and form them into single cell suspension. Then then both OptiPrep and Nicodens gradients have been used to isolate a motor neuron rich fraction from the spinal cord and the hippocampus using either a simple density barrier or a discontinuous gradient. So this discontinuous gradient may additionally provide a partial purification of some other neural cells. So the gradient for the rat hippocampal tissue are 3 or 4 step discontinuous ones and not simple density barriers. So here we have discussed about the method of the density gradient centrifugation discussed by Brewer et al. So here the cell suspension is layered over a discontinuous gradient consisting of 1.029, 1.036, 1.043 and 1.057 gram per ml of nicotins in hibernate A B27 medium. Brewer et al. Were one of the first groups to shift from nicotins to OptiPrep for hippocampal neuron purification using a 4-layer gradient covering a similar density range. Marx et al. And Krebs et al. Reported the use of an almost identical gradient. So the neurons from cortical tissue, hypothalamus and brainstem have been isolated in similar gradient. In all cases, the motor neuron bands shifts towards the bottom of the gradient. So with the help of density gradient centrifugation, we obtain the following layers. So the motor neuron rich gradient is found towards the bottom, that is in the fraction D. So this is the OptiPrep that we use. This is an ideal density gradient medium for the isolation of cells. This is a sterile endotoxin tested solution of 60% iod xanol in water with a density of 1.32 gram per ml. So the high density of OptiPrep facilitates the fractionation of cells from a dense load zone through either a continuous or a discontinuous gradient or through a simple density barrier. So this is the protocol how we do it. So we take the rat or mouse brain or spinal cord, we dissect the hippocampus or the cortex of the spinal cord as the case may be, and slice them, digest them with the help of papine. We triturate or we with the help of scalpel and a needle we mechanically separate it to release the cells. Then we purify the neurons with the help of the density gradient. Then we concentrate the cells and resuspend in a desired medium. So if it is a neurosphere culture, we will culture them in the appropriate medium, neurobasal media, and the B27 minus retinal acetate plus growth factor medium. Or if it is a neuronal cell culture, we will culture them in the appropriate neurobasal and B27 media along with several growth factors. Then we plate them on the appropriate substrate and then incubate and grow them. So this is how the density gradient centrifugation looks like. So these are the several fractions that we have obtained. The fraction 1 we get the oligodendrocyte, and we have the fraction 2, fraction 3 and the fraction 4 where we get the microglial cells. So the fraction 3, this portion is the area where we obtain the neurons, and this is one way by which we can separate our neurons from the other non-neuronal cells or the glial cells. Next, we have FACS, so the fluorescence activated cell sorting method, which is another method by which we can separate our cells. So this is quite a modern technique by the help of which we can separate our cells. So as it is evident from the name itself, fluorescence activated cell sorting, it utilizes three major components, that is one is the fluidics, optics and the electromagnetic. So here we have the cells, as we had discussed in the lectures, that the cell is a dynamic cells are very dynamic in nature. They contain several receptors on the surface. So these receptors are capable of binding to appropriate antibodies, and if we label these antibodies with the help of several fluorescent conjugates, we can separate the cells based on their size, their level of complexity, and also the antibodies or the receptors that they express. So here, with the help of fluorescence activated cell sorting or FACS, we can separate the cells on the basis of the size, the level of complexity or granularity, and the different receptors that they express on the surface. So we have here we have a flow cell that project our cells into a single cell suspension, and here we subject our cells to several antibody, which are then themselves conjugated to several fluorescent dyes. Now say if you have a cell A and you have a cell B, so both of them express different types of receptors on the surface. So now if you have a prior knowledge of the type of receptors that these cells express at a particular time, so then you will be able to target these cells with the specific antibodies. So we subject the cells, we treat the cells with the specific antibodies, which are in turn conjugated with several fluorescent dyes, and each of these dyes will express or they will emit fluorescence in response to different excitation wavelength. So now we separate each of these cells will, as they come out of this region, that is this single cell suspension comes out of this region, each of these cells will be activated with the help of a laser light. So the light could either be scattered in a forward direction, or it could be scattered in the sidewise. So the forward scatter tells us about the size of the cell, whether small or large, and this side scatter will tell us about the level of complexity of the cell. So if it is a highly complex cell, that is a granular cells, as you can see in the case of the white blood cells, each of the white blood cells have exhibit a different kind of complexity within themselves. So with the help of this side scatter, you can determine the level of complexity of the cells. And then we have the fluorescent. With the help of these receptors, which are present on the surface of the cell membrane, uh, you can uh, you can treat them with several antibodies. The antibodies will express several fluorescently labeled dyes, which will give a particular color, and with the help of this, you can separate the cells. So here you can see is evident from this graph. So we plot a graph like this. So along this x-axis, we measure the forward scatter, and along this y-axis, we measure the side scatter. And depending on this, we can isolate our cells. So suppose your cells have are smaller in size, and so just imagine that you have your forward scattering along this x-axis, and your side scattering along the y-axis. So as I discussed before, the forward scattering will tell us whether about the cell size, whether they are small or large. So next we if the cells are smaller in size, so they will be situated more towards the more towards this side, and if they are the larger cells, they will be shifted more towards this side. Similarly, if your cells are highly granular, that is they are very complex, have a very complex structure, so then they will be more towards this upper region, while if they are less granular, they will be towards the this region. So with the help of this, we can understand or determine the level of complexity and the size of different cells, and also separate them on the basis of that. So now we move on to discussing the assignment questions. So question number one. So question number one is astroglia. We have four options here. Maintains cellular homeostasis. Are the immune cells of the brain, form the myelin sheath, or none of the above? So we have discussed about the different types of cells and the glial cells and their different functions. So now you can answer in the chat box what you think the right option could be. So okay, so three of you are saying that it should be option A. So the astroglia maintains cellular homeostasis. So before we move on to discussing the solution, let's discuss about the option B and option C. So uh, are the immune cells of the brain. So as we have discussed that which are the immune cells of the brain. The immune cells of the brain are the microglial cells. Next we have form the myelin sheath. So the myelin sheath is mainly formed by the Schwann cells in the peripheral nervous system and the oligodendrocytes in the central nervous system. So we are left with one option here, that is maintain cellular homeostasis. So let's see what the right answer is. So the right answer is maintain cellular homeostasis, that is option A is the correct option. Now we move on to the next question number 2. So the microglial cells, the maintain cellular homeostasis, are the immune cells of the brain, form myelin sheath, or none of the above. The microglial cells are another kind of cells in the another kind of the glial cells. So these questions are somewhat similar to each other. So again, uh, all of you are saying that it should be option B. Now we will see. Yes. So option B is the right option. Microglia are the immune cells of the brain. So we had discussed already in about the microglial cells in the preceding slides. So I haven't provided you a description uh, of the microglial cells here. So now we will move on to our next question. So question number 3, the density gradient centrifugation utilizes a column with what density, top to bottom. So we have a blank here and 4 options: decreasing, increasing, same, or none of the above. So you can write the answers in the chat box and we will discuss about them. So whenever we are talking about a gradient, it is pretty evident that the density could not be the same. So option C can never be the right answer here. Next we have option B and option A, increasing and decreasing. So even if you are not aware of the right of the procedure of the density gradient centrifugation process, even then, even if you even if it is evident from common sense itself that the density will increase towards the bottom of the falcon or whatever the vessel that you're using, because the center of gravity will pull the heavier objects towards the bottom. So it is evident that the larger cells will be found towards the bottom of the density gradient. And so a couple of you are saying uh, that it should be option B. So let's see what the right answer is. So the right answer is increasing density. Uh, Now we will move on to our next question, the question number 4. So which of these cells are not a cell of the nervous system? The neuron, the astroglia, the Schwann cells, or the nephrons? So the neuron, as we have discussed, they are the cells which are associated with the function of transmitting signal from one neuron to the next across the nervous system. Then we have the astroglial cells, which are mainly associated with providing support to the and growth to the neuronal cells. Then we have the Schwann cells, which are found in the peripheral nervous system and are associated with the formation of the myelin sheath around the axon. And then we have the nephrons. So the nephrons are found in the kidney. They are the basic functional unit of the kidney. So option D is not the correct option. Yes. So option D is the right option. That is, the nephron is not a cell of the nervous system. Now let's move on to question number 5. Which of these is a recommended density gradient medium? Semi-prep, semi-osmo, opti-prep, or opti-osmo? So many of you are saying that it should be option C, opti-prep. So let's see what the right answer is. So we have discussed about this in our previous sections already. So OptiPrep is used as a density gradient medium. Next we have question number 6, which is somewhat related to question number 5. So based on your answer in question number 5, what is the density of the recommended density gradient medium? Option A 0.32 mg per ml, option B 0.32 gram per ml, option C 1.32 mg per ml, or option D 1.32 gram per ml. So you can write your options, the right option in the chat box and we will discuss. So now moving back to our to some of the preceding slides. So we have discussed about this. So this is the OptiPrep, which is used as a medium for the isolation of cells. The high density OptiPrep facilitates the fractionation of the cells uh, by flotation from a dense load zone through either a continuous or discontinuous gradient. So this is we discussed about the protocol, how we do it. Next, here we have discussed about the different densities. So the density gradient is formed from top to bottom. Increasing density we have. So here we can find the answer that it has a density of 1.32 gram per ml. So now we will move on to our um, answer. So the right answer is option D, 1.32 gram per ml. Now let's move on to our question number 7. Density gradient centrifugation is based on only buoyant density, sedimentation rate, both buoyant density and sedimentation rate, or none of the buoyant density and sedimentation rate. So you can write the answers in the chat box and we will discuss. So all of you are saying there should be answer C. Let's see. So yes, answer C is the right option. That is both buoyant density and sedimentation rate is that is density gradient centrifugation is based on the buoyant density and the sedimentation rate. So the primary function of the density gradient centrifugation is to separate particles either on the basis of their buoyancy density or their rate of sedimentation. An ideal density gradient media has the following properties. So these are some of the properties which are the requisite of an ideal density gradient media. That is, they should have the sufficient solubility to produce the range of densities required. It does not form solutions of high viscosity in the desired density range, is not hyperosmotic or hypoosmotic when the particles to be separated are osmotically sensitive. The solutions of the gradient should be adjustable to the pH and the ionic strengths that are compatible with the particles being separated. It does not affect the biological activity of the sample, is non-toxic and not metabolized by the cells, does not interfere with the assay procedure or react with the centrifuge tubes, exhibits a property that can be used as a measure of concentration, can be easily removed from the purified product, is autoclavable, and is of reasonable cost. So these are some of the properties that an ideal density gradient media should have. Now let's move on to the next question. Question number 8. Which motor neurons regulate the size of the muscle spindle or the intrafusal fiber? Alpha, beta, the gamma, or the delta motor neurons? So you can write your answer in the chat box. So three of you are saying that it should be option A. So let's see what the right option is. So the right option is not A, it is option C actually. That is, the gamma motor neurons regulate the size of the intrafusal fibers. So the intrafusal fibers are the skeletal muscle fibers that serve as specialized sensory organs or the proprioceptors. They are innervated by sensory nerve endings in the central non-contractile parts and by gamma motor neurons in contractile ends and serve as a sensory proprioceptor. They detect the amount and rate of change in length of a muscle and constitute the muscle spindle and are innervated by both sensory and motor fibers. Intrafusal muscle fibers are not to be confused with the extrafusal muscle fibers, which contract, generating skeletal movement and are innervated by the alpha motor neurons. So um, the intrafusal fibers are innervated by the gamma motor neurons. So this in the adjoining figure you can see the extrafusal muscle fibers and the intrafusal muscle fibers. Next we move on to our next question. So the question number 9 is also somewhat similar to question number 8. Which motor neurons regulate the size of the extrafusal fiber? Alpha, beta, gamma, or delta? So we had already discussed the answer in our previous slide, the previous question. So you can write the answer in the chat box. Yes. So the right answer here should be A, that is alpha. The alpha motor neurons regulate the size of the extrafusal fiber. The alpha motor neurons are the right answers. Alpha motor neurons must not be confused with the gamma motor neurons, which are associated or innervate the interfusal fibers. The extrafusal muscle fibers are the standard skeletal muscle fibers which are innervated by the alpha motor neurons and generate tension by contracting, thereby allowing for skeletal movement. They make up the large mass of the skeletal striated muscle tissue and are attached to bone by fibrous tissue extensions or the tendons. Each alpha motor neuron and the extrafusal muscle fibers innervated by it make up a motor unit. The connection between the alpha motor neuron and the extrafusal muscle fiber is a neuromuscular junction where the neurons signal the action potential is transduced to the muscle fiber by the neurotransmitter acetylcholine. So you can see in the joining figure, these are the axons of the gamma motor neurons which innervate the interfusal fibers here. Then we have the extrafusal muscle fibers which are innervated by the alpha motor neurons, which is I guess not shown in the picture. So let's move on to the last question, question number 10. So, which clustering occurs on myotubes at the neuromuscular junction? Nuclei, acetylcholine receptor, cytoplasm, or the astroglia? You can write the answer in the chat box and we will discuss. So, we had discussed about the neurotransmitters during our discussion of the nerve impulse transmission across the synapse. So, acetylcholine is one of the examples of the neurotransmitters. There are several neurotransmitters available. It could be either excitatory or it could be either inhibitory. So, the excitatory neurotransmitters are associated with depolarizing the adjacent neuron, the postsynaptic neuron, while the inhibitory neurotransmitters are associated with um, repolarization. So, what kind of clustering occurs on the myotubes at the neuromuscular junction? So, let's see what the right answer is. A couple of you are saying there should be answer B. So, the option B is the right answer. Acetylcholine receptor clustering occurs on the myotubes at the neuromuscular junction. The clustering of acetylcholine receptors at the sites of the nerve muscle contact is one of the earliest events during the differentiation of the postsynaptic region of the neuromuscular junction. So that's all for today's session. I would like to acknowledge NPTEL and IIT Madras for giving me this TA-ship opportunity. Thank Professor Moinak Das, the course instructor for this course, for selecting me for this role. I would like to express my gratitude to my supervisor, Professor Mahitosh Mandal sir. I would also like to thank MHRD, Government of India, for the Prime Minister's Research Fellowship. I thank IIT Kharagpur for allowing me to pursue PhD at the institution. Lastly, I thank all participants for joining the session. Thank you.